everyone can see my screen. Let me know if you can't. All right, so we're talking about how important it is to normalize samples by read depth. And the way that we are going to do this, there are multiple ways to do this, as Amanda reviewed, but the way we're going to do it is using the DSeq2 package, because that's what we're using for differential gene expression analysis. And there is a built-in normalization step. It's the first step in the algorithm, DSeq2 algorithm. And this method is called median of ratios. And we're going to go in detail into the math, even though it's only one line in the R code. So the way median ratios has, I believe, 4 steps. So the first step here is to look at all the samples and calculate the geometric mean across all the samples. So the geometric mean is the square root of the product of all samples. So this is for each gene. And we create what we call a pseudo-reference sample for each gene. So across every row, we perform the geometric mean calculation. We get our value, and that serves as the pseudo-reference sample for that gene. Then we calculate the ratio. We start with the sample. We take the sample over the pseudo-reference for every single gene. And so we're adding a whole bunch of columns here. We have the sample ratio of sample 1 over the reference, ratio of sample 2 over the reference for every single gene. So the rationale here is that because the majority of genes are not differentially expressed in an experiment, that's an assumption that's been empirically demonstrated. The majority of the genes in each sample should have similar ratios within the sample. So by calculating the geometric mean and then taking these ratios, we're going to end up with ratios that are pretty similar. So you can see for most genes, the ratios within sample 1 are close to 0.3. So 0.28, 0.13, 0.39. But then down here, MOV10 is a 0.59. Okay, so that gene is looking a little bit different. Also, if we look at sample 2, the ratios of the first 4 genes are also pretty similar. They're all around 0.7, except for MOV10, which is 1.3. So in this step, we are sort of pulling out which genes seem like they're different from the other genes within a sample. We then take the median value of all the ratios for a given sample, and that's taken as the normalization factor or the size factor for that sample. So the median is now taken down the column. So for example, if we're just looking at the first 5 genes here, the normalization factor for sample 1 will be the median of these 5 values, and the normalization factor for sample 2 will be the median of these 5 values. So the normalization factor, the reason that we're calculating this is because this is the value that is actually going to be used to do the normalization. It's going to be used to adjust the data. So we're actually changing the gene expression values by adjusting them using this normalization factor. And the way that we use the normalization factor to do the adjustment is we take each raw count value that we started with and divide it by that sample's normalization factor. And that then generates the normalized counts value. So, for example, just to bring this down to earth a little bit, if we were to look at the raw counts for these 2 genes in these 2 samples, you'd think, wow, if we look at the expression of ABCD1 in sample 1 versus sample 2, we'd say, well, sample 1 has 22 versus only 13 counts in sample 2, that's almost double in sample 1. ABCD1 must be differentially expressed. But after we normalize using the normalization factor that's calculated using the median of ratios, we then see actually the adjusted values are really similar, 16.92 and 16.88. So ABCD1 is actually probably not differentially expressed. So the normalized counts are giving us more reliable insight. So after we've performed the median of ratios normalization, we can then go ahead and start comparing genes directly between samples, but we couldn't do that before normalization. So given what we've just talked about, if we did not perform read depth normalization, what result would you expect if we compared gene expression values between two hypothetical samples, one with read depth of 5 million reads and one with read depth of 20 million reads? What result would we get in a differential expression analysis? What do you guys think? Right, exactly, Hanley. So we would expect the difference would be significant and they would be differentially expressed. So basically, we would think if we get differentially expressed genes, most, if not all of them, would look like they were overexpressed in sample 2 because more raw counts are simply assigned to each gene in sample 2 due to deeper sequencing depth. So as a researcher, why would this result be problematic? Yeah, exactly, Eowyn. It's not providing an accurate picture of what's actually going on biologically. Which genes overexpression is actually due to true biology and which are due to the sequencing depth difference. Yeah, exactly. All right. So the first step of DEC2, before we can do any differential gene expression analysis, we have to make sure to normalize for differences in read depth, which as you'll note, were simply derived from the data that we gave DEC2. We didn't have to know the read depth of each sample. DEC2 derived that through the median of ratios method. The second thing that we need to do after normalization, we're ready to perform differential expression analysis in our hypothetical iris experiment. That would be around 100,000 hypothesis tests run at once, one for each gene. As we discussed before, DEC2 uses a walled test for differential expression analysis hypothesis testing. And can you guys remind me, what is the null hypothesis that we're testing for each gene? Right, exactly. That this gene is not significantly differentially expressed. So there's one more step that we need to do before we can perform the differential expression analysis. And this has to do with the idiosyncrasies of RNA sequencing data. And this is, we kind of got into this earlier when we were talking about distributions. So what we need to do before running the Wald test is look at the gene-wise variance, which in this case is called dispersion, but it's the same thing mathematically. We learned how to calculate variance. Dispersion just refers to variance. So the thing is, in RNA sequencing data, if you have genes with really low mean counts, it's easy to get technical stochasticity. So if there's just not a lot of that gene to pick up in the cell, it's easy to pick it up sometimes and not other times. And so what that means is that genes that are just have naturally low mean counts, the variance that you get in your data for that gene is often unreliable. We just know that biologically we're not seeing the variance that is occurring biologically just because it's hard to reliably measure genes with low mean count. So these genes could still be interesting though. They're not low. These are genes that are, not low enough that they've been filtered out through our counts row sums less than 10 filter. They're just low enough that the variance is unreliable. So they're probably still interesting to us. And so DESeq2 helps us out by solving this problem through estimating what the actual variance for this gene should look like by sharing information across genes. So if you think about all the genes in a data set, the variance distribution of each gene is going to be similar across samples as long as those genes have similar expression levels. So DESeq2 is assuming that genes with similar expression levels are also going to have similar variance. So DESeq2 then just shares information across genes with similar expression levels to impute or estimate the correct variance for genes that have lower mean counts. And this is important because we talked about how the Wald test establishes mathematical models and how mathematical models are just rules for the data. Well, these rules often have a lot to do with variance. So we talked about how we use basic mathematical functions to build the models, including mean, variance, standard deviation. So we really need to be able to rely on our variance estimate. Felix says, I wonder if the filtering step to get rid of the fewer than 10 transcript genes was to improve the variance estimate. That's a really good point, Felix. I don't know off the top of my head if that's why that step was designed, but I think that's a pretty good hypothesis. I'm sure it didn't hurt either. Because I imagine that if DESeq2 were trying to share information across genes that are super noisy because they're really lowly expressed, this step wouldn't work so well. The estimation of variance. It's a really good point. So, right, so the reason we need to have reliable estimates of variance is so that the Wald test can make mathematical models that are reliable. And the way that DESeq2 does this estimation is using a statistical process called maximum likelihood estimation. So just in regular terms, this means that the most likely estimate of dispersion is calculated for each gene, given the gene expression values from all the replicates in each group. So its maximum likelihood estimation is pretty intuitively named. We are just saying, what is the most likely variance or dispersion for this gene, given all the other genes and all the other replicates in this group? So after dispersion has been estimated for all of the genes with low counts, we can go ahead and run our mathematical model. We can pick a mathematical model, run a Wald test. So as we talked about before, the Wald test creates a mathematical model and identifies which model, the null hypothesis or the testing hypothesis, best maximizes the likelihood of the data. And as we were talking about before, the type of model that the Wald test uses is dependent on the shape or the distribution of the data. Because the mathematical model, along with the mean and variance of the data, has to know what is the distribution shape of the data. Otherwise, we talked about how the t-test is a little bit less specific than the Wald test because it just relies on mean. Well, the Wald test takes into account what does your data look like when you plot it out. So the Wald test has to be told which general type of distribution to use, which is great. That makes it really flexible. You can use it for RNA-seq data, tell it what distribution is right for RNA-seq data. And for other types of data, you can tell it a different distribution. So the Wald test is really nice for RNA-seq data because RNA-seq data has a specific shape, and we can specify the Wald test to match that shape. Because just the nature of RNA sequencing data is that the majority of genes end up having an expression value close to 0. When you plot out all the expression values in an RNA sequencing data set, there's a really long right tail because there's also no upper limit for maximum expression value. Because we're just measuring gene counts, we're just asking the cell how many counts were there for this gene. It can be all the way up, we saw, to nearly 200,000. But the reality is most genes are not that highly expressed. Most genes have expression value close to 0, including before and after the filtering step. So because RNA-seq data has a long right tail, we need to pick a specific distribution for that. Sometimes count data is modeled with the binomial distribution, which can be illustrated by the probability of, good point, Felix, getting ahead a little bit of me. So count data is sometimes modeled with the binomial distribution, which can be thought of as the coin toss distribution, or it can be sort of illustrated by imagining the probability of getting heads when you toss a coin a certain number of times. So for example, in this graph, if you toss a coin 40 times, your probability of getting heads is about half of the time. But not all count data can be fit with the binomial distribution because this distribution is really only useful in situations where there's a relatively small number of data points. Obviously, we have thousands and thousands more than 40 data points. So we use the negative binomial distribution. The negative binomial distribution looks like this, and you will notice that that is very similar to what you would expect for RNA sequencing data. So it makes sense for you to use this distribution. Felix is absolutely right that this does look like the Poisson distribution as well. There's a specific reason why you don't use the Poisson distribution here, and if you will just bear with me for 1 second, I will look it up. I originally had the Poisson distribution in these slides, but it seemed like a little bit too much material. Okay, so just to show you this quick slide, as Felix noted, there is another distribution that does look very similar to the RNA sequencing data distribution, long right-hand tail. But the limitation of the Poisson distribution is that the Poisson distribution is assuming that the mean and the variance are the same. And this is often not true for gene counts data. So the negative binomial distribution is specifically appropriate for data where the mean tends to be less than the variance. And that's what we see. I haven't really talked about this, but this is a known characteristic of RNA sequencing data where the mean of your data set is much less than your variance, right? So you can imagine the mean for this distribution here is going to be somewhere way back here, closer to zero, whereas the variance is all over the place because of this long right-hand tail. So the negative binomial distribution is specifically the most appropriate distribution for RNA sequencing data due to this idiosyncrasy that there's this long right-hand unbounded tail in the distribution. So great point, Felix. Thank you. Oh, I guess I am actually at the end of what we're going to talk about in the lecture right now. The next part is, the last part of the lecture is visualization, but we're not going to do that until we do a little bit more coding first. So I would like everyone to please open up their Jupyter notebook again. And since I'm assuming that we've all mostly disconnected from the San Jose State VPN since yesterday, we're probably going to have to reopen our RNA-seq DGE notebook. So please go ahead and do that. And once it's open, please make sure that the kernel says R idle or R busy in the bottom left-hand side. So that's the RNA-seq DGE notebook. Please let me know if anyone is for some reason having issues connecting to the Jupyter notebook, Jupyter Hub. All right. Now, since we have closed the kernel, we can't pick up where we left off until we rerun everything that we ran yesterday, because we defined a bunch of variables, we imported a bunch of libraries that we're going to need to use today. So please go ahead and run through all of the cells that we ran yesterday, all the way through the end of section 1. So that's the part where we wrote out our DE-seq dataset object. And just as you're running through, examine the outputs to make sure you're not getting any weird or unexpected errors. Oh, good point, Rashi. You can select a specific cell. That's true. If you want to, you can scroll down to our last run cell, which was the right.csv with the DE-seq dataset object. You can go up to run and run all above selected cell. I am just going to keep doing this since I started it. But if you do Rashi's method, just make sure you scroll up and make sure that you didn't get any errors. Yeah, exactly. You guys are experts. All right, so please let me know in the chat if anyone saw any errors or is having any issues. I'm going to slowly move on to step 2, which is where we are going to implement what we just learned about in the statistics lecture. So we have talked about how each sample has different read depth and the issues that the differences in read depth can cause if we think that a sample has higher gene expression, but it really is a technical artifact from sequencing depth. This can affect the interpretation of our downstream results by tricking us into thinking that we have statistically differentially expressed genes biologically, when really they're just technical artifacts. So what we can do to visualize this this scenario is to make a Pca plot and see what it looks like before we do any normalization at all. So just to recall that we call Pca dimensionality reduction method because it's taking our high dimensional gene expression data and condensing it down into principal components or vectors that capture the main sources of variance in the data set. Can you remind us which step we're on from the flow diagram at the top of the Jupyter notebook? Sure. So we have started with raw counts, and we've made and sample metadata, and we've made our DSeq dataset object. So we're now going to make a PCA with raw counts. Yeah, no problem. Okay. So the first thing before we actually perform our visualization is that we need to transform our data using a log2 adjustment. So we are just taking the log of all of our raw counts, and remember that we are adding 1 so that we don't end up taking the log of 0, because that gives us a negative infinity text in our data frame, and then that's going to throw an error when we try to feed it into something that requires a numerical matrix. So we take the log because we noticed the huge spread in values all the way from 1 all the way up to 20,000. And if we do a log transformation, that brings the data into the same ballpark, so it's easier to visualize. So we're going to take the log of our raw counts data frame and then pass that into a variable called exp_raw. Then we're going to use a new function called prcomp, which computes the principal components of our raw expression. I do want to note here that somebody yesterday, I forget who, mentioned that in machine learning tends to put variables as the column names, whereas here our variables, our genes, are the row names. Well, in this case, the prcomp function expects features or variables to be columns instead of rows. So we first need to transpose our data frame. We need to flip it so that the rows are columns and the columns are rows, using a handy function in R called T, which stands for transpose. So the output of computing our principal components is going to go into a new a variable called Pca_raw. So let's go ahead and run this cell and then take a look at the principal components that we've calculated using the summary function. So let's take a look at this output together. How many principal components were detected? Just take a look at the output and let me know what you think. Yeah, 12, exactly. So we're looking at a table here, which is being printed out on kind of on 2 pages, where each column is one principal component, and there are 12 columns. What about taking a look at the rows of this table? What percent of the variance, remember each principal component takes into account or summarizes a certain percentage of the variance of the data set. What percentage of the variance is explained by PC1? And then what percentage of the variance is explained by PC2? Yeah, exactly. So the proportion of variance row tells how much of the variance is explained by each principal component. PC1 explains about 18% of the variance, and PC2 explains about 13.6% of the variance. Now, finally, how are these principal components ranked? Yeah, in descending order, exactly. So the in descending order of specifically the proportion of variance explained by each principal component. So as the variance explained by each principal component goes down, we sort of walk down the table here. So each subsequent principal component explains less and less of the variance. So no, Rashi, that's actually a good point. As I was answering this question myself, I noticed it's not, it's not just proportion of variance. Proportion of variance is descending. Standard deviation is also descending. And then cumulative proportion is actually ascending. So that's because all three of these metrics are related to each other. So as variance decreases, standard deviation also decreases. We're just particularly interested in the variation here because this is a proportion of variance. So it's set up in terms that are easy for us to understand. What percentage of variation in our data set is explained here? But no, that's a good point. Yeah. All right, so let's go ahead and plot the first two principal components against each other in a scatter plot. We're going to use a function called autoplot, pass in our PCA results. We're including our sample table, which remember shows which genes are flight and which ones are ground control. We're going to color the plot by condition, which is the column name in sample table that shows flight and ground control. In this plot, we're going to label all the samples so we can see where each sample falls in the plot. And we've specified the size of the label, the size of each scatter plot dot, and then alpha, which is the transparency of the points on the plot. So let's just go ahead and run this cell, and everyone should see a PCA plot that looks like this. You can see our ground control samples are green, our flight samples are red. Keep in mind, this is the raw and unnormalized data. So we are taking a look at this plot for illustration and educational purposes, but you wouldn't want to make this plot for publishing or anything unless you were specifically comparing it to your normalized plot, which we will make in a minute. Note that the percentage of variance is shown on each axis, which is really nice. And let's go ahead and use another function called ggsave to save our plot to our Pca directory with a nice informative plot name. Now we can go ahead and create exactly the same plot and save it, but without the sample labels by passing false to the label argument within autoplot. So let's do that, see what that looks like. I think this is cleaner, but of course there's less information. Phil notes that the color argument is spelled using British spelling. If you spell it with the American spelling, the argument is not recognized and you get a default black plot. That is a really good point. Thank you, Phil. Must be British developers. All right. So also, for going forward, super annoying. Yeah. All right, so now that we've kind of established how do you label or not label your plots, we're just going to show the plots without samples labeled, but you can go ahead and change it back to label equals true at any time if you want to see which samples are which. And before moving on, let's just go ahead and save the principal component values into a CSV table. And we're saving that also into our Pca directory. All right, so taking a look at this plot, let's answer a question. Is there a pattern in how these samples are separated? What do you guys see here? What's the pattern? What do you mean by that exactly, Hannah-Lee? Like you can draw a line that separates. That's true. You can draw a line that separates them. Let's think like a biologist or a bioinformatician. What is going on in terms of the biological conditions? Eowyn says flight has more of a spread. That's absolutely true. James says the most variation is between the two conditions. Right. So imagine that you ran this experiment. The reason that you ran this experiment is because you were asking what is the difference between mice that were flown on the ISS and mice that were kept on the ground. So you are really interested in the difference between these two conditions. So when you look at this plot, you really want to see, is there a difference? Do I see a difference between the two conditions? And you absolutely do. There's a huge gap. As Hanley pointed out, you can actually draw a line to separate the flight from the ground control. And if you look at so if you look at the x-axis represents the first principal component, which explains almost 20% of the variation in the data, and the separation between the two conditions is along the first principal component. So most of the variation in the data is the variation that separates flight versus ground control. So this is a nice result for someone who is interested in the difference between flight and ground control. Now, as Eowyn pointed out, there's a big spread in the flight samples. That, we say, is the separation along the second principal component, which explains 13% of the variance in the data. So the spread within conditions is being captured by the second principal component. All right. Any questions on all that information on the Pca plot? So let me rephrase that, Eowyn. On this particular plot, so I wasn't saying something that can be broadly assigned to all PC1 and all PC2. I'm just saying when I'm looking at this particular plot, I'm seeing, when we say, oh, 20% of the variance is captured by PC1, I'm like, okay, what does that actually mean? What variance? To me as a biologist, what within the data set is causing that variance? Well, when I look at this plot, the difference between flight and ground control appears to be causing most of the variance in PC1. Then if I take a look at PC2, I say, well, the samples are not separating by flight and ground control condition along PC2, but I do, along PC2, I do see the spread within the flight samples and the spread, a little bit of a spread within the ground control samples. So whatever is causing, and we don't know this, whatever is causing the flight samples to kind of spread out a little bit, that's the variance that's captured in PC2. All right, good. Okay, now we are going to go ahead and actually perform the size factor estimation or normalization that we just learned about in the lecture. And here we need to, we're going to run all three DSeq2 steps. So we're going to do size factor estimation, we're going to estimate gene dispersions, and we're actually going to run the Wald test. Here we need to submit a script with commands for all three steps to the Slurm job queue. And to do this, Felix was asking, how do you run a Unix command in a notebook running an R kernel? You use the system command, and we're passing to the system function a string which has the command that we want to have run on Unix. So sbatch and then the path to the Slurm script. So let's go ahead and run this, and then we're going to walk through the actual code that we're running within the Slurm script. So we've gotten, once we get this started, we can go through and look at the code. So size factor estimation is, remember, correcting for sample-wise differences in read depth. We're going to use a function provided by DSeq2 called estimate size factors. And this function performs all those steps that we talked about, calculating the median for each gene, taking the ratio of each sample, all these things. That's all packaged in this function. And the way that we run this is by calling the function, passing to it our DSeq dataset object, and we're assigning the output to an updated DSeq dataset object that we're calling DDS_1. So super easy to just normalize your data using size factor estimation. Very straightforward. Secondly, remember, we need to estimate gene dispersions because some, the genes with the low counts don't have reliable gene variance or gene dispersion. So we have to share information between genes. Again, we have a very handy function provided by DSeq2 called estimate dispersions. We call this function and pass to it our updated DSeq dataset object, and then we just write the results back. We overwrite our updated DSeq dataset object here, so we just continue calling it DDS_1. There's another function called plot disp ests, where we can visualize the relationship between the gene dispersions and the mean expression of each gene. So this kind of visualizes what we do when we estimate the gene dispersions. So I've just put this plot in here so you can take a look at it. Um, the um, the let's see, questions here. Yeah. So here, what we're looking at is a plot with the mean expression of each gene on the x-axis, and each dot here is a gene. The dispersion is on the y-axis, and the black dots represent genes prior to gene estimation, and the blue dots estimate the final estimates. And the red line is the fit line that was used to estimate dispersions. So taking a look at this plot, what is the relationship between gene dispersion, which is here on the y-axis, and mean expression of a gene? So what do you see? What happens to, for example, mean expression as gene dispersion goes up? Any ideas? There's a negative correlation between the mean and the dispersion up to a point, then it kind of plateaus. Yeah, exactly. Nicholas, as the dispersion goes up, the mean goes down. Or as the mean increases, the dispersion goes down. So this is the second part of this question is, what does this tell you about RNA sequencing data? This is what we were talking about in the lecture, where as the expression of a gene decreases, the variance goes up because the measurements for a gene with really low mean expression are unreliable. So the variance is kind of just going all over the place. Yes, Felix. So the red line is the line to which the data are being fit. So as DESeq2 is estimating the dispersion for each gene, it's bringing them all closer to this line by looking at the trends across the data. The black dots are the values of the genes prior to estimating dispersion, and the blue dots are after estimating dispersion. So, yeah, no problem. So the second question here is, can we say how the gene expression data are changing before and after the gene dispersion fitting or the gene dispersion estimation? So what's happening here between the black dots, which are before dispersion estimation, and the blue dots, which are after? What's the general trend here going from black dots to blue dots? How are the gene expression data changing? Smaller values look like they increase more than larger values. Yes. Um, yes, exactly. Because and dispersion overall is going up. But what's happening is we're bringing a lot of these genes, which are sort of hanging out here below the main distribution, bring them up closer to the fit line. And in particular, the genes that have a lot of very high variance are being brought together. So the dispersions are being estimated and they're being transformed to a fit line. Felix, that's a really good question, and I don't know. I would have to look into the if there are options for this built-in function to just show the fit line. So I can look into that and get back to you. But are you thinking that would make it easier to sort of interpret what's going on here? Yeah, let me look into that and get back to you. All right, so we can come back to this. But in the meantime, let's go ahead and move into the third step where we actually start doing the hypothesis testing, or the differential expression analysis using the Wald test. So again, there's a really nice built-in function called n-binom Wald test. So this is a function that specifically runs the Wald test using the negative binomial distribution, which we saw before is the most appropriate distribution for RNA sequencing data. Let me make my text bigger here. So we talked in the lecture about the t-test and the Wald test, but is the Wald test the only hypothesis testing method that we could have used here? Go ahead and let's click on this link and look at the likelihood ratio test section here. So DSeq2 actually offers two different kinds of hypothesis tests. And so we've alluded to this before when we were developing the contrasts. We only have two comparisons here, right? But the Wald test is fine with that. The Wald test just compares two groups together. The likelihood ratio test, on the other hand, is really nice when you have three or more levels of a factor or three or more groups. And so in a situation where we would have multiple conditions, for example, flight, ground control, maybe drug treatment, non-drug treatment, or something like that, we would use something like the likelihood ratio test, which will compare all of our different levels of factors. So the reason that this is important to note is because the GeneLab DSeq2 differential expression pipeline, the standardized pipeline, runs both the Wald test and the likelihood ratio test, just to cover the cases where there are multiple groups or factors that need to be considered. In this case, this is a little bit of a simplified case where there are only two groups, so we're only running the Wald test. But just to note, there is another type of test that we can run. So why do we choose the Wald test? Because we only have two groups. All right, so at the end of our Slurm script, we just exported our normalized counts as a data frame. is we ran the median of ratios normalization, and then we wrote out a CSV with the normalized counts, and we also wrote out the sample table so that we can use it later. So let's go ahead and see how the normalization with median of ratios changes the global gene expression. So PCA is a really nice tool for looking at how does the global expression across the data set change. First, what we need to do is read back in that normalized counts table that we wrote out during the Slurm script. So we're just going to use the read.csv function and read in the normalized counts table, read it into a variable called norm counts. And I'm just going to add a cell here so that we can take a look at what we're seeing here. I have to spell it right. So if we run head norm counts, we can see we've adjusted all of the gene expression values in the matrix. They're all different than they were in the raw counts table. Our columns are still sample names and the rows are still gene names. So let's again log normalize our, or log transform our normalized counts table and run prcomp to calculate the principal components. And go ahead and use autoplot and ggsave to create a Pca plot without labels and save it. So why don't you guys compare, scroll up and down, compare this Pca plot to the Pca plot that we generated prior to normalization. And what are you seeing? Is the percent of variance explained by PC2? Is it more or less after normalization? Yeah, exactly. It's less after normalization. So before normalization, PC2 is accounting for 13% of the variance. Now PC2 is accounting for 11% of the variance. So what does that tell you about, so all we've done is we've normalized by sequencing depth. So what does that tell you about what was contributing to the differences or the variance within PC2? Excellent, you guys, exactly, read depth. So, Zinru, what do you mean by outliers exactly? So, because the variance in PC2 decreased, I'm not sure. Okay, let me tell you how I think through this, and then maybe it'll become a little bit more clear. So, because the variance in PC2 decreased after we normalized for read depth, we can say that some of the variance in PC2 was due to differences in read depth across samples. Not all of it, because we still have, 11% variance, but it's gone down, so that's good. I forgot the normalization is used on read depth. Yeah, exactly. Now, take a look at the within group variation. So the variation within the flight samples and the variation within the ground control samples. How has that changed? Yeah, less variation within the ground control group. Um, why do you think this is? So the answer to this question might be a little bit more straightforward after we talk about the third question. How is the percent of variance explained by PC1 changed? Yeah, exactly. It's gotten bigger. So before normalization, PC1 was 18%. Now it's about 20%. So this is what I think of as a kind of a nice side effect of normalization, where when we normalize, we're trying to get rid of technical variation. Well, the variation in the data set also includes real biological variation that we want to be amplifying and pulling out and separating from the technical variation. So as the technical signal has decreased, what do we see? We see that the biological signal, the difference between flight and ground control, which is the separation across PC1, that has increased. So as we're getting rid of the noise, our signal is going up. And that's exactly what we want after normalization. So if we look back at question number 2, how has the within group variation changed? We see, ground control samples are tightening up a little bit. We also see the flight samples seem to be pairing up, which is a little bit interesting. We're not sure why that is, but if we're talking about seeing increased biological signal after normalization, it's possible to hypothesize that some of this tighter structure here may be due to biological signal coming through. But again, we're not sure. These are hypotheses we can generate based on the Pca plot and then and investigate later. All right. So in the last 5 minutes before lunch, go ahead and use the next 2 code blocks, and feel free to make more code blocks, to first of all, go ahead and list the number of PCs after normalization, see if they were the same as before normalization, and then go ahead and recreate the Pca plot. But adjust some parameters. So adjust the label size. You can put label back to true. Adjust the size of the dots, and then you can play around with alpha. But keep in mind, alpha has to be between 0 and 1. So alpha is a measure of transparency. So see how that changes. And you can go ahead and copy that auto plot command from the previous code block. Okay. Nicholas, I was just saying that in the last couple minutes before lunch, you guys can play around with the auto plotting command. All right, so we'll pick up here after lunch, and if you guys noticed anything interesting with your plots or had any errors, we can go over them then.
Hey guys, before you take off to lunch, I just want to let you know, let me go ahead and stop this recording.